Hi everyone, welcome to Dento'ZEN. We are continuing with our topic of morphology and physiology of bacteria. This topic we have already discussed classification, types of microscopes, different staining methods and various shapes of bacteria in our previous videos. You can check those videos. We have also discussed various structures in bacterial anatomy, and today also we are going to discuss one important topic here, that is bacterial spores. And in our last video we'll discuss the growth and multiplication of bacteria. So let's start with bacterial spores. Before starting, quickly subscribe to Dento'ZEN, tap on the like button and hit the bell icon so that you remain notified about new videos. So under bacterial anatomy, we discussed the structure of a normal bacterial cell. It is made up of outer layer or cell envelope, protoplasm and cell wall appendages. Then we also discussed the various atypical forms of bacteria which can be important short note for your exam, and we discussed it in the last video. And today we are going to discuss bacterial spores, which can be an important short note for your exam, or there can be so many entrance or viva questions from this topic. So let's get to know what are bacterial spores. Now, bacterial cell, whenever it is under unfavorable conditions, for example, there is excessive heat, there is desiccation or drying, there are chemicals, there are radiations, or there is depletion of nutrients, less nutrients. So what this bacterial cell will do? This bacterial cell wants to protect itself, so it will form a structure within itself which will have multiple layers, and those layers will protect it. So this structure is called bacterial spores. So bacterial spore, as you can see, it has multiple layers. So this structure is highly resistant structure, highly resistant to all those factors, and it is a resting or dormant stage of bacterial cell. That is, bacterium becomes metabolically inactive here in this structure. So spore is a resistant, highly resistant, resting or dormant stage of bacterial cell formed under unfavorable conditions. Now, when the conditions become favorable, what will happen? Now, when everything is fine, this spore will go back to vegetative cell. It will form a cell again. So this process of formation of spore from the bacterial cell is called sporulation, and the process of formation of cell from the spore is called germination. Something is germinating, something is forming. So those are the two processes: sporulation, formation of spore, and germination, formation of cell from the spore. Now let's see how to write for your exams. So bacterial spores are highly resistant structures, highly resistant resting or dormant stage of bacterial cell. They are one of the most resistant forms of life, and they can remain viable for centuries. For so many years they can remain like that. So they are formed under unfavorable conditions, and they are formed due to depletion of nutrients. Now, as these spores are forming within the bacterial cell itself, within the cell, so they are also called endospores. Endo means inside, so they are forming inside the cell. Now, there are certain specific genera of bacteria which commonly show spore formation, and that you have to remember, that is Bacillus and Clostridium bacteria. They commonly show formation of spores. So that is very important for your entrance exam. Which bacteria most commonly associated with spore formation? Bacillus and Clostridium. Now this spore, as you can see, it has multiple layers, and it is made up of calcium inside it, and several layers are there. So we need to know the structure of this spore and the layers. So let's see the layers one by one. So what can you see in the center? In the center there is core. Core is the center. The center or the core contains the DNA, genetic material. Then there is the membrane around it, that is inner membrane, and there is a wall which is called germ cell wall. So that is the core. So this inner membrane helps in forming the cell wall when this spore will turn into cell again. Now after that, there is another thick layer, that is cortex. Cortex protects it, and it has outer membrane. Then there is one more tough layer, that is called coat, as blue one, as you can see here. And some bacteria or may not have another layer, that is called exosporium, that is the outermost, which may or may not be present. And this layer is a loosely placed layer, and it has some ridges over it, and this may help in the attachment of the bacteria. So we have seen the layers, that is core containing DNA, inner membrane, germ cell wall. Then we have the cortex, which is present between the inner and the outer layers. And then we have coat, which is a tough layer made up of proteins and which provides additional protection to the spore. And then in the last, we have exosporium, which may or may not be present. So you can remember CCC, core, cortex, coat. Now, what is sporulation? It is the process of spore formation. But how does this spore formation start? First, it starts within the bacterial cell. Within the bacterial cell, at one area, at terminal area, a clear area forms. As we can see here, a clear area starts forming. This clear area becomes opaque, and then it is called forespore. And then this forespore will develop these layers and will become a mature spore, as we can see here. So this is a mature spore with all the layers. Now this mature spore will give rise to one cell. So one spore forms only one cell when conditions become favorable. So we can say that sporulation is not the process of reproduction. It is not the process of division. And it is not a method of reproduction, and it is a complex process. It may take about 10 hours, and mature spore that is formed is extremely resistant to heat and disinfectants. Now, what is germination? Germination is when the spore will again give rise to bacterial cell. So transformation of dormant spores into active vegetative cells when they are grown in nutrient-rich medium. So that is germination. So we have discussed what are bacterial spores, what is sporulation, and what is germination. Now let's see what can be the shape and position of spores. Within the bacterial cell, for every species, this position, shape, and relative size are constant. So let's see what can be the different positions of the spore. So let's see, this is a bacterial cell, black one. Within it, red one is the spore. So it may be located in the center of the bacterial cell. So this is called central, or it may be located in the subterminal position, that is just near the terminal, just near the end, that is called subterminal, or it may be located at the end or the terminal, and then it is called terminal position. So similarly, if we look at the size, sometimes this spore may be bulging. So it is called bulging, central bulging, subterminal bulging, or it may not be bulging. So then we can call it central non-bulging, as we can see here, or subterminal non-bulging, as we can see here, and then terminal oval. So based on the shape, it can be terminal spherical, as we can see the shape here, or it can be terminal oval. So based on the shape, it can be oval or spherical. Based on the width, the diameter may be same or less than the width of bacteria. And then the sporicidal agents, that is agents which can kill the spores. So spores are resistant to most disinfectants. Sterilants can kill spores, and the most commonly, most effectively used sterilant is autoclave or ethylene oxide sterilizer. So that can be your important entrance question. The most effective method of killing the spores is autoclaving. Now, how do we see these spores? How do we demonstrate? They have so many layers. In the gram staining, they appear as unstained refractile bodies within the cells. They remain unstained, and when we see them in unstained sections, they can appear as refractile bodies. Then they are also weakly acid fast, so we can see them with modified acid fast staining or modified ZN staining, where they can appear red. And then we have the special stains for them. The most commonly used special stain is Sheffer-Fulton stain, and then we have also another stain, that is Moeller stain. Now in Sheffer-Fulton stain, we can use two dyes, malachite green and safranin. So the bacterial cell will take up the safranin and will appear red or pink, and the spore will take up malachite green and will appear green. Then we get to know the applications of these spores. Now they are resistant to so many things, so we can use them to check if the sterilization is proper or not. So they can be used as indicators of proper sterilization in different sterilization techniques. For example, in autoclave and plasma sterilizer, spores of Geobacillus and Styrothermophilus can be placed within them, and after sterilization we can check if the spores are there or they have been killed. Then second is spores of Bacillus atrophius can be used for the sterilization control for hot air oven and ethylene oxide sterilizer. So they can be used as indicators of proper sterilization. Another use of spores can be as agents of bioterrorism. That is, that means some people can use these spores to cause damage to humans. So these spores, especially the bacillus spores, the bacteria bacillus spores, can be used for this purpose. So those are the demonstration and applications. Let's quickly summarize. So spore is a highly resistant structure. It is a dormant stage or resting stage of the bacterial cell forms under unfavorable conditions. So the process of formation of spore from the bacterial cell is called sporulation, and it begins with the formation of a clear area within the bacterial cell at its one end, which then later becomes opaque and is called spore, and later it develops layer and it becomes a mature spore. Now this mature spore has multiple layers. See, starting from the center it has core, which is made up of DNA, inner membrane, germ cell wall, then cortex, which is in the middle, and then coat, which has outer membrane coat, and exosporium, which may or may not be present. Now, based on the different positions and sizes and shapes, they can be central bulging, central non-bulging, subterminal non-bulging, or terminal spherical or terminal oval. So those can be the different arrangements of the spores within the bacterial cell. Now, sporicidal agents which can kill the spores can be sterilants, can be autoclave or ethylene oxide. Autoclave is the most effective method, and then the methods of demonstration staining is gram staining, where they appear as unstained material, or modified ZN staining, or special stains, that is Schaefer, Fulton and Miller stain. And then their applications, they can be used as indicators of proper sterilization and agents of bioterrorism. So that is how you can write for your short note. Now let's check what have you learned. So first question, process of spore formation is also called germination, conjugation, sporulation or fission. Then question number 2, a bacterial spore is a reproductive cell, highly resistant, dormant structure under stress, or bacterial flagellum type, or a toxin. Then question number 3, which genus is most commonly associated with spore formation, Staphylococcus, Streptococcus, Bacillus or Escherichia coli. And finally, an effective method of killing bacterial spores is alcohol, pasteurization, autoclave or boiling water. So that is all for this video. If you really enjoyed the video, do tap on the like button and share this video with your friends. Keep watching, keep learning, keep smiling. See you in the next video soon. Till then, take care. Bye bye.